Okay, this last video segment is about assembling your nucleotides and making your model. So I'm just going to show you the things that you're going to need to begin with. You're going to need a little thing of glue. You're going to need a darning needle, which you can get from the supplies on the counter. You'll need a bag of the pre-cut straws in a Ziploc bag. You'll need your nucleotides, of course. You're going to need a washer, and you're going to need your piece of floss, which you'll find over on the counter as well. Okay, so the first thing I'm going to do is to quickly tie on my washer to the bottom. That's going to help you to, as you assemble it, to kind of give it a little bit of weight and for when you start to glue it. Okay, and then at the other end of my string, I'm going to need to thread my needle. Okay, and give yourself quite a bit of length there so that it makes it a little bit easier. All right, this is going to go pretty fast. What you want to do is to read your code. So remember, I've got my sample one here. Yours is going to be on your function finder paper. And what you want to do is to find your first letter. And remember that we're using the nucleotide that's on the right side as our key. That's the one that would be, this would be a C paper. And what I'm going to do is I am going to place a straw on my, thread my needle through the straw. And then I am going to poke through this hole. There's no hole, there's a little black circle. I'm going to poke through that with my needle and thread it through. And I have put on my first nucleotide. I'm going to grab another straw. Okay. And now I'm ready for the next one, which would be an A. So here's one of my A nucleotides, and I'm going to poke that right through there, thread it through. And sometimes the thread, of course, wants to kind of get in the way. I'll just kind of carefully get it going there. There we go. All right, next, another straw. So in between every nucleotide paper, you want to have a straw. Okay, and then I would just keep going here until I get all of them threaded on. All right, now I'm not going to do all of that. I'm not going to do all of that. Bring that back up here, because that's going to take a while. But what I am going to do is to show you how they start to get glued together. And I'm just going to have these three here. Something about the DNA molecule that's really nifty is that it makes a spiral as it goes up or as it goes down, kind of like a spiral staircase. So I'm going to be able to start to orient the papers, and I'm going to start here on the right side, and I'm going to place a little dab of glue here on my tab. That's that flat tab that was going up. And this OH here that was flat on the one right above is going to sit right on top of that blank space. And I'll be able to line up those lines from that phosphate to show that those bonds are there. It's going to hold it there for a second. Okay, then I'm going to do that for the next one. It works really best if you have someone else in your group, another partner, member, holding it, and they can kind of hold it up and they can up high so that the other person can be working on gluing. Okay, hold it there for a second. Okay, now when I hold it up, you can see that notice how it's starting to turn. Okay, they're not going to be like stairs on a ladder that are right above each other, like rungs on a ladder. It's starting to twist. That's the whole thing that you want to have happen. That's an important part of the model. And if that's not happening, then you're not making the model correctly. Okay, I'm going to kind of spin it a little bit so that I can do the ones that were going down. Okay, and notice how I have this flat area on the phosphate. I'm working now kind of downward. I'm going to place a little bit of glue there. And the nucleotide that was down below that, I am going to put those, layering that on top there, gluing it, holding it for a second so it'll stay. And then I'm going to do the same thing here. And notice again, this is this flat white part here. That part is where the glue goes. And that H and OH is going to sit right on top of that. Hold it for a second, kind of let it set. Okay, now I'm just going to show you this. This is obviously incomplete at this point, only has 3 nucleotides on it, but notice how it's already really getting that spin to it. When I look at it from the side, you can see how the base pairs are the flat part and how the phosphates and the sugars are the pieces that are going up on the right side and going down on the left side. Okay, you're going to have 36 of these in a sequence. It's going to get really long, and you may need to have your partner even maybe stand up on a chair to kind of hold it for you. And you'll end up with them all threaded with your code in that code order that you have on your paper, and then you'll have completed your DNA model. Make sure that you put your sticky note label back on your model, or label, even better, label a nucleotide or two so that we're sure that we can identify your particular model. All right, there you go.